Just a quick recap, we talked about the electron transport chain. We compared it to the power plant, the hydroelectric power plant that produces electricity for a whole country, the whole city, and all of that. The same way our cells produce energy for the whole body, for the cell itself, and for the whole body through the electron transport chain in the mitochondria. We describe the process as a chain of events that occur in the mitochondria. In the inner mitochondrial membrane, you have a series of complexes that transfer electrons from one complex to the other. We talked about the chemiosmotic theory or chemiosmosis that is coupled with the electron transport. That means that as electrons are moved from one complex to the other, there is free energy that is released that causes the pumping of protons from the matrix to the intermembrane space. And this causes a gradient which eventually causes oxidative phosphorylation, which is simply the addition of phosphate to ADP, which produces ATP. We talked about the mechanism of that, so if you want, you should go back and check it. We are moving straight to the next part of our discussion on the electron transport chain, and we are talking about the sites of oxidative phosphorylation. Here we are seeing three major sites of oxidative phosphorylation. Remember, we have mentioned that oxidative phosphorylation is basically the addition of phosphate to ADP to produce ATP. But before that ever happens, there are a lot of things that need to happen, which we have already mentioned. The transfer of electrons to the last carrier, the transfer of electrons to the last acceptor, which is oxygen, and then at the same time, the pumping of protons into the intermembrane space. So sites of oxidative phosphorylation does not necessarily mean where the phosphate or ATP is produced. It basically means those sites that contribute to the production of this ATP. Because ATP will not be produced if those protons are not pumped out. And we are seeing that these sites are the sites that cause the pumping of protons into the intermembrane space. We see site 1, which is NAD and coenzyme Q. So when NAD is giving its electrons to coenzyme Q, there is a pumping of protons. Site 2 is cytochrome B to cytochrome C1. And cytochrome B in complex 3 is giving its electron to cytochrome C1 still in complex 3. There's a free energy that is released that causes pumping of protons into the intermembrane space from the matrix. The same for site 3. So at the level of complex 4, cytochrome A and cytochrome A3, the movement of electrons till the final acceptor of electrons, which is oxygen. This particular movement causes the pumping of protons, so it releases free energy that is able to pump protons from the matrix into the intermembrane space. So these are the sites that are able to cause oxidative phosphorylation, because what we saw is that if the protons are not moved out of the matrix into the intermembrane space, there will be no gradient that causes them to move back in, and they move back in through ATP synthase, which causes phosphorylation or oxidative phosphorylation. So that's basically it. All right, we mentioned before in one of the pathways that we are going to see the value of NAD and FAD. Their value means we mentioned that they are going into the electron transport chain to produce ATP for the body. How many ATP molecules will one NAD eventually produce when it gets into the electron transport chain? We have already seen the major function of NAD. NAD brings the electrons, FAD also brings electrons. But we are seeing that NAD also contributes in pumping of protons. So at the end of the day, NAD produces 2.5 ATP molecules per cycle. Or when one NAD molecules comes into the electron transport chain, eventually 2.5 ATP molecules are produced. When one FAD comes into the electron transport chain, 1.5 ATP molecule is produced. You might be asking yourself, is there half an ATP molecule? What do you mean by half ATP molecule? When phosphate adds to ADP, basically you produce a full ATP. How can you produce half ATP? But let's not get worried here. We'll explain it. So one ATP molecule is produced when 4 protons pass through ATP synthase. So 4 protons that were pumped into the intermembrane space are responsible for producing one ATP molecule. So how many protons are pumped out into the intermembrane space when NAD comes in? you see that there are 10 protons that are pumped into the intermembrane space, because NAD itself, which is complex 1, site 1, causes the pumping of 4 protons, site 2 causes the pumping of 4 protons, and the last one causes the pumping of 2 protons. So altogether you have 10. And then when FAD comes in, remember FAD is in complex 2, and complex 2 does not contribute any pumping of protons. That's why you have minus 4 protons, which causes it to produce less ATP than NAD. So basically, if 4 protons cause 1 ATP to be produced, it means that when NAD comes in, it produces 10, it causes a pumping of 10 protons. So eventually 2 ATP molecules will be produced, and there will be 2 protons left over. There will be 2 protons left over. And those 2 protons that are left over would eventually be filled up by the next NAD that comes in. So it's not like the body is going to produce half an ATP molecule. No, it's just telling you that there's a leftover of protons, which will definitely be filled up by the next NAD that comes in. The same for FAD. There is a leftover of 2 protons, which will be eventually filled up by the FAD, the next molecule of FAD that comes into the electron transport chain. So basically, that is it. So with this knowledge of the value of NAD and the value of FAD, you can be able to calculate the total number of ATP molecules that a cell will produce when it breaks down glucose, for example, from glycolysis, and then eventually goes into the electron transport chain and then to the ETC cycle. So you can try to do that calculation, the total number of ATP molecules that are produced when one acetyl-CoA, for example, goes into the TCA cycle. How many ATP molecules will be produced? I want the answer in the comment section. All right, to the final topic in our ETC cycle, to close the electron transport chain, we'll do as we do for most other pathways. We talk about regulation, we talk about inhibition. But in the electron transport chain, we're going to be talking about inhibition and uncoupling. We have seen inhibitors before. When something inhibits another thing, meaning it prevents that thing from happening. While now we are adding another word, uncouplers. We mentioned along the line that oxidative phosphorylation, for example, is coupled with chemiosmosis. Chemiosmosis is coupled with electron transport. Coupling means two things happening at the same time. Uncouplers will definitely come in between that couple. Okay, so basically they'll stop those two things from happening at the same time. So uncouplers will probably block ATP or oxidative phosphorylation from happening, but it will not have anything to do with the transport of electrons. It might not alter anything along the line of transportation of electrons, or maybe the pumping of protons, but it will stop oxidative phosphorylation from happening. ATP will not be produced, while inhibitors will block at the level of the transfer of electrons. So it will block the complexes, it will inhibit, it will stop the complexes from carrying out their function of transferring electrons. So we see a series of inhibitors and uncouplers here, but we're not going to go into details for all. We're going to mention a few. The first one we're talking about is amobarbital, which is a pharmaceutical. Basically, it is used in treating mood disorders, for example, like depression. But this amobarbital is a complex 1 inhibitor. So it inhibits the movement of electrons to coenzyme Q. So if it inhibits that, eventually electrons will not move to the rest of the chain or to the rest of the components of the chain, and eventually ATP will not be produced at all. So inhibitors block the progress of the chain. So eventually there's no continuation of electron transport. And of course there will be no ATP produced. But what we'll see that uncouplers do is that they do not have anything to do with the movement of electrons in the chain. By the end of the day, ATP is not produced. What will be produced with uncouplers is heat. So instead of ATP being produced, there is an increase in heat production. All right. So we talked of amobarbital. We have rotenone. Rotenone is found in the roots of pea plants or leguminous plants, but it's also found in high quantities in pesticides, for example, insecticides. Okay, for complex 2, we have carboxyl, we have malonate. Malonate is basically an industrial, found in industrial waste products or industrial products, and malonate is a complex 2 inhibitor. Carboxyl is a fungicide and is basically used by farmers to prevent the seed from being attacked by fungi, or yes, by growing, when you harvest corn, for example, and it starts growing mold, there are some uh, chemicals that are used or fungicides that are used, and they contain carboxin. Complex 3 inhibitors, we have antimycin A, we have British antileucite. Antimycin A is produced by Streptomyces bacteria, but let's talk about this British antileucite. Basically, another name for dimecapal, which is an antidote for, it's usually used as an antidote for metal poisoning. So if you consume a substance, a chemical that contains some metals, they will give you this dimecapal, and it is supposed to bind to those metals, chelating them. That's how they call it in chemistry, chelation. Bind to these metals and you're able to eliminate it. But the hand British antileucite can also be an inhibitor of complex 3. And then you have complex 4 inhibitors. Basically, they inhibit the transfer of electrons from complex 4 to oxygen, which is a final acceptor. And examples are cyanide. Cyanide is a common poison and is found in food like cassava, which is, but especially in cassava that is not sweet, the one that is quite bitter or too hard. There's a high amount of cyanide in it, which is quite toxic, and it is toxic by inhibiting complex 4. But processing of the cassava, for example, when they fry it or cook it or do whatever, reduces the amount of cyanide in it. Carbon monoxide, which is produced by incomplete combustion, that's chemistry. But for example, the smoke that comes out from car exhaust contains carbon monoxide, and it is also a complex 4 inhibitor. Hydrogen sulfide, basically from waste, that gas that has that rotten egg smell, from sewage. It could also be from industrial waste. It is a complex 4 inhibitor. And then we have ATP synthase inhibitors. Remember, ATP synthase is the enzyme that eventually causes oxidative phosphorylation to happen, or ADP to be converted to ATP. But ATP synthase can also be blocked or inhibited by some molecules like oligomycin atractyloside. And then we have the uncouplers that we mentioned. The ones that are starred are physiological uncouplers. Physiological means that they are found naturally in the body. So thyroxine is a molecule that is found in the body. LCFA, long chain fatty acids, they are gotten from food, but they can also be synthesized in the body. And then we have thermogenin. Thermogenin is basically an uncoupler of the electron transport chain. It's physiological, yes, but another name for it is uncoupling protein 1. So it's found in brown adipose tissue. Brown adipose tissue is found mostly in newborns and in animals that are hibernating. So remember, we said that with uncouplers, uncoupling causes the ATP not to be produced, but what is produced instead is heat. So newborn babies and animals that are hibernating, when you're hibernating, animals hibernate usually in very cold conditions, and when babies are also born, they don't have this protective, their skin is not as protective as ours, so they have brown adipose tissue to be able to break down or to be able to carry out electron transport, but instead of producing ATP, they produce heat instead, because when animals are also hibernating, they're not doing much, so the body does not need as much ATP. And children too, they're not working anything, so basically they don't need that ATP. And so the brown adipose tissue helps the breakdown of, or this cycle to end up in the production of heat, which is protective for them also, and also helps in maybe protecting the cells from producing excess ATP when it is not needed. Bilirubin, for example, is usually produced from breakdown of red blood cells. So if it's in excess, it will cause uncoupling. We have the other ones which you can check out. Those ones are not physiological, so they are definitely found in probably chemicals or waste products or industrial molecules or industrial chemicals.